I'm very happy to introduce to you Sam Miller. Sam joined the U.S. Geological Survey's Virginia and West Virginia Water Science Center in 2021 as a hydrologist. His research focuses on watershed scale practices that influence downstream hydrologic responses across urban, agricultural, and natural environments. He's particularly interested in how human activities affect water quality and ecological health. His current projects include regional and national studies on pesticides and per and polyfluoroalkyl substances, PFAS, as well as investigations into factors influencing aquatic macroinvertebrate health in the city of Roanoke, Virginia. So we welcome Sam. Take it away, Sam.
Hello, my name is Sam Miller. I'm a hydrologist at the USGS Virginia and West Virginia Water Science Center. I'd like to thank this conference for letting me present recent research that my colleagues and I have completed. In this study, I'll show you how we applied a wastewater model to predict pesticide concentrations in the Potomac River Basin. And then using a sampling campaign, we validated the model and identified dominant sources of pesticides. And then finally, we'll wrap up with results from a more recent study looking at long-term imidacloprid trends from a national network. But before I get started, I just want to acknowledge all the help I got from my outstanding team of collaborators. It was a real honor and privilege to work with such wonderful scientists. I also want to acknowledge the USGS programs that funded this research. So I'll first present how pesticides can be sources of wastewater, and then introduce our accumulated wastewater model. And then I'll show you results from this model that were recently published in 2 journal articles in which both pesticides and PFAS concentrations were predicted from accumulated wastewater. And then finally end with this more recent national scale trend study in which 2 sites are from within the Potomac River Basin. So the USGS has been detecting wastewater contaminants in streams for decades. This includes things that maybe you've thought about before, such as pharmaceuticals, hormones, antibiotics. Certainly nowadays, PFAS is on everyone's radar, but pesticides can also be found in wastewater. On the right, I'm showing one of the largest wastewater treatment plants in the world that's within the Potomac River Basin. I know for myself, I often take these really impressive feats of engineering for granted. They turn our raw sewage into treated effluent, which can be returned to our surface water bodies, which in the modern water cycle is a very important component. However, you can imagine removing contaminants at very fine trace concentrations, such as parts per trillion, is really challenging. So it's important that we can, understand where wastewater is located and characterize potential chemical exposure. So pesticides can be introduced to municipal wastewater from a variety of ways. Perhaps more obvious, we've detected pesticides in tap water and in human urine. Various laundry and cleaning products contain pesticides. There's a growing attention to pet, flea, and tick treatments as a potential down the drain source, as well as numerous industrial processes that may have direct discharges to municipal wastewater. So if we can develop these wastewater models to estimate contributions from point sources, together with measurements, we can identify, whether point sources or non-point sources are contributing to contamination that could be targeted for management. So that's what we did in this study. We've been developing this accumulated wastewater model that builds off a decade plus of research that started with this drinks model, in which Rice and Westerhoff estimated the percentage of treated wastewater in drinking water intakes. And on the lower right, you can see the new and improved modern water cycle, in which the emphasis on human use is really emphasized. And I'm circling the municipal and industrial water use. This is an integral part to the modern water cycle, not only in arid regions of the world, but in places like the Potomac River Basin, such as the Occoquan River, which was one of the first potable indirect water use projects in the world, in which we have a drinking water intake downstream of a very large municipal wastewater treatment plant. So it's important that we can, understand where wastewater is located and predict potential chemical exposure and effects to human and aquatic life from wastewater. So Rice and Westerhoff collaborated with USGS authors to develop this model for the Shenandoah River watershed, a sub-watershed of the Potomac. And in that study, basically, they identified where municipal wastewater treatment plants are located, these white circles, and how much discharge they have into stream segments. And then with that information, we can calculate the accumulated wastewater percentage and take that one step further and predict contaminant concentrations. That study was recently expanded to include the entire Potomac River Basin and included both municipal and industrial wastewater. And as I mentioned, they predicted various concentrations from different classes. And so the current studies pick up where this left off, where we're now attempting to model more types of pesticides as well as PFAS. So as I mentioned, we conducted a comprehensive monitoring effort during the summer of 2022 during low flow conditions when accumulated wastewater percentages would be highest. As you can see in this map, we collected samples from all over the basin, from the forested headwaters to the developed coastal plain. We sampled sites with absolutely no wastewater to sites that were a majority accumulated wastewater. In addition to pesticides, we sampled for PFAS, pharmaceuticals, trace elements, major ions, dissolved organic matter characterization, really to get a holistic view of the water quality. And then we compiled lots of data from literature on treated effluent wastewater concentrations and decided to focus on these 14 target pesticides. These were pesticides that were reported most often in literature and frequently detected. And so our model input concentration represents the median value that accounts for the non-detect percentage in parentheses. Now we also modeled using a hypothetically lower and higher concentration to acknowledge the range of uncertainty in this input data set. So on the right, you can see the circles represent municipal wastewater treatment plants scaled by their discharge. And so we assume that the effluent coming from these plants was representative of our model input concentrations. So now looking at what was detected, represented in these colored box plots compared to the aquatic life benchmarks that are these small symbols. First, I'm showing you everything that was detected, and I'll kind of zoom in on the target pesticides. 49 pesticides were detected. The insecticide imidacloprid and herbicides atrazine and prometon were detected at almost all the sites. Almost all the sites had at least one detection. However, the average number of detections per site was 19 different pesticides, for a cumulative concentration of about 400 nanograms per liter on average. Also wanted to highlight that 10 of the detected pesticides are technically PFAS within some jurisdictions of the Chesapeake Bay, meaning that they have a fully fluorinated carbon atom. This includes the insecticide fipronil and its degradants. So yeah, the presence of this strong bond may allow these chemicals to persist in the environment for longer periods. All right, so zooming in on these target pesticides, fortunately, most of the detections were orders of magnitude lower than any aquatic life benchmarks. However, I will note that 2 insecticides frequently exceeded these benchmarks, imidacloprid as well as fipronil. So now showing how our measured MINTs compared to our predicted concentrations. we can see that there's a variety of fit denoted by the Spearman correlation depending on the pesticide. However, most of the predictions using the median input represented by these colored symbols is below this diagonal one-to-one line and points to other landscape or non-point sources. The smaller triangles represent hypothetical predictions using higher or lower model input concentrations, but on average, our predictions were about 10% of our measurements and highlight the role of other non-point sources. So to look into the non-point or landscape sources, first we looked at this really valuable data set that the USGS produces on estimated agricultural use. We found that agricultural use of pesticides was significantly correlated to the herbicides, atrazine, metolachlor, simazine, and insecticide clothianidin. However, in general, most fungicides and insecticides were more related to indicators of developed land use. And this is challenging because we do not have any accurate non-agricultural pesticide use data sets to further evaluate spatial patterns. However, you can see from our data that we detected significantly higher concentrations of fungicides as well as insecticides in developed watersheds. So we really wanted to kind of tease apart this developed versus wastewater signal. And we found that our ancillary water quality, primarily this organic matter characterization, was particularly helpful. So using this principal component analysis, you can see in this wastewater quadrant, we see things like the accumulated wastewater ratio, at the optical brightener signal, which is an organic matter characterization. You see things like the pharmaceutical concentrations. Whereas in the developed quadrant, we see things like developed land use, refined fuels as an organic matter characterization, more indicative of just general urban runoff. We see that the herbicide simazine was more closely related to agricultural use, and a few pesticides didn't really show any particular dominant use. However, I should emphasize that all these pesticides have both agricultural and non-agricultural use. And it just happens with our data set that these were the dominant sources identified. And it's possible in other parts of the country or Chesapeake Bay, you might find different patterns. So to evaluate potential risk to invertebrates, we calculated a cumulative toxicity quotient. We did that by dividing the measured concentrations by the chronic benchmarks for all detections at each site. And so a value over 1 indicates potential risk to chronic exposure. And you can see on the y-axis, this extends far beyond 1, and the majority of sites appear to be at risk to chronic exposure. This is primarily driven by the presence of imidacloprid as well as fipronil bifenthrin. And I just want to highlight this important study that came out last year from USGS that synthesized a decade of studies that concluded that there's multiple lines of evidence that point to pesticides as stressors to invertebrates in several regions of the United States. When we think about our impaired streams, many of them are impaired because they lack healthy invertebrate communities. They require management plans and implementation plans that do not address pesticide contamination when present may fall short of these efforts to improve these communities. So real quickly, I want to highlight the results from our PFAS measurements. Overall, we see a significant positive relation between accumulated wastewater and total PFAS measured. However, there's some notable exceptions pointed out. These sites all have presumptive PFAS sources, such as airports, fire training facilities, military sites, landfills, as well as just urban watersheds that have a history of urban chemical use, including pesticides. And while we worked on the PFAS and pesticide study independently, I just want to highlight that these chemical classes often are occurring in mixtures. And I encourage you to check out some of the previous recordings from this conference that highlight the role of PFAS and pesticides. And PFAS are added directly and indirectly to our pesticides for their surfactant properties. The US EPA detected high concentrations of PFPEA, PFHXA, and PFBA in the rinsate of these fluorinated containers that contain pesticides. In Maryland, studies have detected high concentrations of PFAS in mosquito spray. So there's a potential that the application of pesticides could be a source of PFAS into the environment as well. So for more information on these studies, I encourage you to read the papers, but if you're more of a visual learner, we've created these interactive maps to explore the results of our model. So in this map, you can zoom around and click on different circles, which represent stream gauges and wastewater outfalls. And so on each stream gauge, you can see how the stream flow compared to the accumulated wastewater ratio, and toggle different layers to look at things like predicted pesticide concentrations or PFAS concentrations. All right, now to shift gears and talk about our recent trend study on imidacloprid. By many accounts, this was the most popular insecticide used in the world in the last couple decades. This is a systemic neonicotinoid insecticide that's used in both agricultural and urban settings. In agricultural settings, it's commonly used as a seed coating, but also as a spray and in granule form. And in non-agricultural use, there's quite a large number of uses, including turf grass, gardens, termite deterrent, invasive species control, forestry, and veterinary products. Imidacloprid is known to be highly soluble and moderately mobile, meaning that there's a potential for leaching into groundwater where it may be able to be transported large distances. And in previous studies, they've found that actually a pretty small percent of the active ingredient of these seed coatings is actually taken up by plants, leaving the majority of the active ingredient available for transport. Of course, neonicotinoids are concerned to insects, in which they're highly toxic to these non-target species, including bees. There's also a growing concern that neonicotinoid mixtures may have harmful effects to aquatic insects like macroinvertebrates. And in previous regional and national assessments done by the USGS, they found that imidacloprid is often driving the potential toxicity of aquatic invertebrates. So now to look at some of the trends from the local Potomac sites. First, one of the sites is this Accoton Creek that I mentioned before. It actually had the highest concentration in the national network on average. This is a small developed watershed that I mentioned had rather high PFAS concentrations as well. This is a site that is impaired due to poor macroinvertebrate health. The concentration doubled over the 10-year period, and almost every sample had a detection of imidacloprid that exceeded the chronic benchmark. Now, this is a watershed that's been studied heavily decades ago in previous assessments and was found to have some of the highest pesticide concentrations in the nation. And now stepping out a bit and looking at the Potomac River, this is a much larger watershed representing a mixed land use. And this watershed had one of the largest percent increases over the study period, approximately tripling over the 10 years. And we calculated that over a ton of imidacloprid was delivered to the Chesapeake Bay from this site over the 10-year period. And taking one step further back, looking at the national scale, we see that almost half of the samples had detections of imidacloprid, and both urban and agricultural watersheds were at risk. I should highlight that many of these white symbols represent sites with very few concentrate detections. These are primarily undeveloped watersheds where humans are likely not using that much imidacloprid. But in sites where we are using this chemical, almost half the sites are increasing and very few sites are decreasing. This includes small watersheds and quite large watersheds. including sites on the Mississippi River, where the concentration approximately doubled over the 10-year period. And we predicted that 143 tons of imidacloprid were transported to the Gulf of America over the 10-year period. So I hope you see the value in these accumulated wastewater models for predicting contaminant mixtures. However, in our study, we were only able to predict concentrations from municipal wastewater, although we know that there are certain industrial sources that may be discharging higher concentrations of pesticides. Aquatic life is often subject to multiple stressors, but the regulatory framework requires us to address the most probable. And at this point, there's multiple lines of evidence that point to pesticides as stressors affecting many regions of the United States. However, in general, lack of chemical monitoring data really inhibits our ability to evaluate risk at larger scales. I just want to highlight in our Potomac study, we measured imidacloprid 2 ways using this conventional mass spectrometry method, as well as this cost-effective immunoassay method, this ELISA method, and they showed strong agreement. This might enable more comprehensive monitoring efforts. This method, ELISA, was used recently in a data release from the Delmarva Peninsula, in which 100% of the samples showed detections. So the widespread presence and increasing levels of imidacloprid, not only in the Potomac River, but across the nation, signal a relation between land use and water quality that may be presenting an ecological problem, but also presenting an opportunity. I'm hopeful that through monitoring, modeling, and management, we can work together to conserve our water resources for future generations. And with that, I'd like to thank you for your attention and happy to answer any questions.
All right. Well, that was Sam Miller from USGS giving us a presentation on their innovative predictive model that helps us estimate the amount of pesticide that is originating from wastewater sources so that we can balance that with other potential sources, all leading to hopefully informed management actions. Wow, really found that imidacloprid data to be compelling. We haven't necessarily had that near the top of our list with regard to occurrence and risk in the past, but the theory that Sam presents here that neonicotinoid pesticide could be impacting benthic communities sounds very plausible. So some important data on the presence of imidacloprid.